Hi guys, my name is Janie Pitta and I am the Events Chair for YSO. This video is going to cover genetic engineering. So what is genetic engineering? So genetic engineering is just the manipulation of DNA to alter an organism's characteristics. So it uses different lab-based technologies to alter the DNA makeup of an organism. So it just adds a gene from one species to an organism from a different species to produce a desired trait. So for example, um, genetic modification is used to make foods healthier. So golden rice, for example, contains a vitamin that's used to make carrots orange. And with the use of this, a lot of people who don't usually have access to this vitamin now do. And these are known as GMOs. Okay, so the steps of genetic engineering. So first you have isolation, which is basically when DNA pieces are first taken from an organism, and then that gene is inserted into the DNA of the host cell. So for this, you need a vector to transfer a gene, and usually a vector is a plasmid, which is like a circular piece of DNA in bacteria. So after that, you have the growth step, which is when your host cell contains recombinant DNA, which is just DNA made from two different organisms, and then your cell begins to multiply. And then after this, your desired protein has been produced. So the first technique of genetic engineering is artificial selection, which is a process when humans or other organisms just consciously choose which traits they favor in other organisms. And when they receive offspring with those desired traits, they work to maintain them. So there's three different types of this, which is selective breeding, hybridization, and inbreeding. So the first one, selective breeding, is when organisms with desired traits choose to mate with organisms with those certain traits. So for example, like broccoli and cabbage were all derived from this wild plant through selective breeding. Hybridization is when two organisms with unlike characteristics are put to produce the best in both. So this is when like matings of two parents from different species happen. And then inbreeding is when breeding with an organism that's like genetically similar to maintain traits. So for example, like mating between like cousins or siblings would be an example of inbreeding. Okay. So the second technique is cloning, which is just the creation of it allows for the creation of like multiple genes. And so the steps in this are, so first a cell is removed from the parent organism, and then the entire organism grows from that cell. So each cell is going to have the same DNA, but the cells are going to vary because different genes are activated for each cell. So for example, Dolly was the first like sheep mammal that was cloned from an adult somatic cell. So her birth proved that specialized cells could be used to create an exact copy of the animal that they came from. And then the third technique is gene splicing. So DNA is cut from one organism and it's inserted into another. So for example, human insulin genes can be removed and put into into a bacterial cell, and this bacteria will now produce human insulin. And genes are cut through restriction enzymes. So these are just enzymes that cuts the DNA at a specific code. And usually gene splicing is used to produce functional proteins. Um, the fourth technique is gel electrophoresis, which is just used to compare DNA from two or more organisms, and it's used for various purposes. And so the process, so first DNA is cut into fragments with the restriction enzyme, and then that cut DNA is put into a machine which is like filled with gel, and then the DNA is like squeezed through those gaps in the machine, and then the machine just separates the fragments based on size. Okay, so next I want to talk about gene therapy, which is just a field of study. So gene therapy is used to correct any defective genes to cure or prevent a disease in your body. So again, restriction enzymes are used to cut the dangerous genes, and viruses are usually used to insert helpful ones. Okay, so pros and cons of genetic engineering. So the pros, they usually help to like tackle diseases, produce new foods like GMOs, and they help us live longer as well. And cons could be that a lot of people don't think they're really ethical because of the way it's done. And sometimes there could be like genetic defects, and it also limits diversity. So let us know what you think, and thank you for watching.